about the thyroid hormones, okay? Thyroid hormones. We studied the thyroid hormones earlier. Is it what type of hormone it is? It is a water soluble or water insoluble, or membrane soluble or membrane insoluble thyroid hormone. Remember that? When we studied the steroid is just like steroid hormone. Okay, it's not a peptide hormone, but it acts like just like a steroid hormone in its membrane solubility. Okay, it is a membrane soluble, meaning it will have what type of receptors? Is a membrane receptor or cytosolic receptors when it's a membrane soluble hormone, because it's membrane soluble so that it can soluble easily and then get into the cytoplasm, so it will have a cytoplasm receptors, so they are floating around in the cytoplasm. Okay, and when this thyroid hormone is going to secrete more, it is whenever there is a lowering of body temperature. Like cold body temperature is lowering. Okay, also if it is increased stress, if you have stress, increased stress. Okay, example, you can also during the starvation, also starvation. All these conditions that lead to what? Lead to increase output, increase of thyroid hormone activity. Meaning thyroid hormone, what type of hormone it is being secreted or increased from the thyroid hormone is going to be thyroxine. Okay, you follow? Thyroxine hormone. So we want to study about the thyroxine, what is the type of molecules are there, we will see now. Okay, this thyroxine or thyroid hormone, it is an amino acid derivative. Amino acid derivative. What amino acids here? We are talking about the tyrosine. Tyrosine is an amino acid, hydroxy amino acids. What is the structure of these amino acids here? It's something like this, and then you have CH2, CH, and NH3, this ammonia molecule, and acid. And this is thyroxine, and it has hydroxy group. So what happened? This tyrosine molecule is combined with the another ring here. Is a derivative, because of this instead of this hydrogen, you have another benzene ring is being attached to it. Okay, here. So it's been removed, this one. So directly attached with an oxygen. I put it like that. This entire thing is an oxygen instead of hydrogen is being removed from here. So this tyrosine molecule attached with another one, and now this molecule is called with the iodinator. Iodine is being added. Okay, iodine.
Dr. Solo,
Yes.
what is that molecule on the far right besides the CH?
COOH. COOH here. This one. Acid group. COOH group. Okay. I put it right. COOH. Okay. So it is being attached to it. So carbon is having 4 valency, right? This is this one valence is CH2, another valence is NH3 ammonia group, another one is hydrogen, and another valence is being with acid group. So it is an amino acid. And this amino acid initially it was a tyrosine molecule, it's hydroxyl group in the para position. The hydrogen has been replaced by another benzene molecule, and the iodine is being added. Capital I with iodide. So this is called, we call it as a T4 because of the 4 iodine molecule attached to the tyrosine molecule, or a derivative of thyroxine molecule, T4 thyroxine molecule. Okay, if the iodine molecule is not 4, if it is 3, sometimes just I put a dotted line. If this is not going to be present, only 3 iodine is going to be there. So if you have 3 iodine molecule, then it is called T3 thyroxine. Why I'm saying this is that T4 and T3 is present in the blood or in our body, it secretes, but the T3 is more active form. Is more active form. You follow up to this? Okay? So now, we'll see. Where this thyroxine molecule is being produced, or this hormone now. As I mentioned before, it is a membrane soluble and similar to steroids, okay? And they secreted from thyroid gland. Thyroid gland, you will see later, thyroid gland. And it secretes as a formation of the follicles. Where and how it is being synthesized. I'll just draw a figure, a follicle, and it is have a lumen here, okay, and these are the cells, okay, right. Now, this is the lumen as the middle, okay, and this here, this iodine molecule is being added to it. Lumen, the iodine molecule should be added. But the iodine is coming from the outside of this follicle. How it is transported across? There is an active transporting mechanism which is existing over there, where it can directly come into the lumen, iodine. To facilitate this process, as I mentioned, is an active transport. You have a co-transporter mechanism. This co-transport, okay, and this co-transport is being combined with another transporter with the sodium potassium. Where the potassium is coming in, K plus, and the sodium is going out, sodium, okay, and the presence of ATP, that's of the molecule. ATP has been energy which is being used to transport the sodium potassium pump, or the transporting process. At the same time, the iodine molecule will be getting into the lumen. So this is also a co-transporter. Transport of iodine. Now, the thyroglobulin, thyroxine, is produced as a TG. Okay, that is, I write it like that, thyroglobulin, a protein molecule which is being produced, thyroglobulin, and it has 660 kilodalton. This is the molecular weight. What is that? Thyroglobulin is a protein, and that is produced with a molecular weight of 660 kilodalton. This is the molecular weight of this protein. This thyroglobulin is nothing but 100 residues of tyrosine. The tyrosine molecule derived, and also iodine is added to it, and thereby it will produce the thyroxine hormone. and it all takes place in thyroid gland. So as I want to produce more of thyroxine, for that what I need two things. One is tyrosine molecule and another one is iodine molecule. So in the cells produced most of the tyrosine residues in the form of thyroglobulin protein. You understand that? Because I mentioned before, this thyroxine is an amino acid. So amino acid, again, if the cell synthesizes a large amount of protein, I mean the protein consists of 100 residues of tyrosine, then the cells will produce this protein and then is exported into the lumen. And there you get the breakdown of these tyrosine residues from thyroglobulin will reduce residues to tyrosine, and the iodine is added together, and then your thyroxine hormone is being synthesized at the lumen here. Okay. You follow now how this thyroxine molecule is being produced? Yes? Or you don't follow now? Okay. How it will happen now? Just I'll write it right here. Large one. Suppose these are the cells which I mentioned before, okay? Okay, these are the nucleus, and nucleus is there. Nucleus, and nucleus is having DNA, gene, and everything, right? And I mean that is being activated, so you get the thyroglobulin. That protein is being produced, and as soon as the protein is produced and is being exported into the lumen. And then on another side, you get the iodide molecule, iodine, which is being passing through the co-transporters and then active transport with the ATP and sodium is coming here. Okay, iodine is there, now thyroglobulin is there. And then this process of thyroglobulin to, from the cells into the lumen by exocytosis process. That we already discussed about it, right? Synthesized in the cells and moved to the lumen. And then lumen, here, lumen iodide molecule is added, and here this iodide molecule, this is going to be added to tyrosine derived. For that we need hydrogen peroxide. It's a H2O2 process which will oxidize this, and then you should add it to it. So now the iodine is added, and it been added in the form of TGI, iodide thyroglobulin, okay, released iodine. And now the next phase is to these things, because these are the tyrosine residues, is attached to the protein. So we need a proteolytic cleavage of this protein. Okay. So what happened? The TGI, thyroglobulin iodide, which has been added to it, thyroglobulin, has been acted by proteases enzyme. Proteases. Okay. And if the proteases is being acted to this, so what will happen now? You get T3 and T4, and these thyroxine hormone, T3 and T4, which I refer together as thyroxine hormone, and now that is released into the blood. Released into the blood. And in the blood, as I mentioned before, it cannot exist as it is, because it is just like a steroid hormone, so it will have a binding protein, okay, otherwise called blood carrier protein, or thyroxine binding protein, thyroxine binding protein. Now it is being carried throughout the body. Do you follow now how the thyroxine is synthesized and how that is released and how that is formed in the follicle and in the lumen, and then how they processed by the proteases? Because the proteases and these enzymes is coming from these lysosomes that will cleave this peptide. As I mentioned before, these are the thyroxine residues with 100 peptide, 100 tyrosine molecules are there, and they are attached with the thyroxine. I mean iodine. Here, here, here. So this is a complex. It's a polypeptide chain, and this polypeptide chain is being added. This is thyroglobulin with the iodide. With the iodide is attached to it. So when the proteases come, it will cleave. It will cleave the peptide linkage like this. See. So what the result is you have 1, 2, 3, 4. So you have a T3, 4. And then if it is cleave only 3, then it will come on, 3 iodine in one molecule, that is a T3. So once this is being broken down into bits and pieces, you get large amount of T3 and T4 will be produced, and that is being pushed into the blood, and from the blood you have a blood carrier protein this name of the blood carrier protein, we call it thyroxine binding protein, which will just like a boat, and this protein, the thyroxine protein, and where the T3 and T4 sits together, bind together, and that is being transported across the body. You follow? Yes? How thyroxine is being synthesized, carried out, and exported into the body now. So now, what is the function of this? The regulation now. The release, there's a regulatory process for this, okay? How this is being controlled. How this T3, T4 is being released. This, there are two processes here. You have to look into hypothalamus in the brain. Hypothalamus in the brain. We are talking about T3 and T4 release. Okay, the conditions. That's what we are going to discuss now. Hypothalamus in the brain. Okay. So in the hypothalamus, if you think about just like I make a drawing like this, and you have a blood capillary is flowing through like that. It comes out like that. Blood capillary is coming. Okay, and also blood capillary. Okay. Now, another part is, this is the pituitary hormone. You know what is pituitary hormones which is there? Pituitary in the brain? Yes? Okay. A cross section of a pituitary hormone. So hypothalamus is the part of this brain here. This hypothalamus, this side. And then there are two parts of the pituitary. Here we call it anterior pituitary, called anterior pituitary. And then here you get the posterior pituitary hormone. I'll just write it here. P-I-T-U-I-T-A-R-Y, pituitary gland, pituitary, where you have the hormones, the pituitary hormones. So the blood, which is coming here, capillary, okay, blood. Then here also blood is coming in. Okay. There are some cells which is in the anterior. Here, cells. Okay. What happened to these cells? They have their own receptors, these cells, the receptors. Okay. Mechanism of release we are talking about it, okay? In the hypothalamus, always they have a neuron. Do you remember that neurons? Yes? And these neurons will have their own dendrites and axons and cell body like the nucleus. And this is the neuroendocrine. And this is being attached with the another axon, another neuron. Okay? So as soon as the signal, because the hormone release, which is being the initial stage where it is being activated in the just mere thought or any other activity onto the brain, and if you have, there is a need, if you're stressed or anything, so the hypothalamus, and they sends out the signal, hey, I want more of thyroxine molecule, what shall we do? So that's the signal which will transmit across to the neuroendocrine cells, and here you get the releasing hormone. The releasing hormone which is coming from neuroendocrine cells or the nerve cells. Okay. it secretes these are the neuroendocrine glands, and this neuroendocrine, these hormone releasing hormones getting into the blood, is coming in, okay, from the neuron. Once it comes out, it will go across to the nearby cells, the pituitary cells, where you get a receptors attached to it. So in the receptors, it will go and bind, and then it will activate, okay, the secretion of another hormone here. That's on the here comes up. Pituitary hormones they produce. So it stimulate the secretion of the neuroendocrine cells, and these neuroendocrine cells in turn it will activate the pituitary, anterior pituitary hormones, and where it will produce mostly on the thyroid stimulating hormone, TSH. Thyroid stimulating hormones, TSH will be there. Not only that, these neuroendocrine will also stimulate growth hormones, follicle stimulating hormones, and TSH and ACTH, and prolactin, and luteinizing hormone, LH. Okay, so all of them which is being activated in the anterior pituitary. You follow that? How this anterior pituitary is a resource of lot of stimulating hormones. It is present in the brain. So the hormones, they got the signals from the neuroendocrine, and thereby it synthesized these hormones. Here we are now concentrating on the TSH now, thyroid stimulating hormone, which is coming here. Another one which I mentioned before as a posterior pituitary. The posterior pituitary, and there it's mainly the neuroendocrine cells, endocrine cells, and they will produce oxytocin and ADH. Okay. So these hormones will also coming from a stimulus from the neurons. Again, here. And it's being attached within the hypothalamus region. So this will depends upon the cells, okay? It will produce this hormone release here, and these hormones also released in the posterior neuroendocrine cells. They also put forward here oxytocins and ADH. This is coming over here. So we will see the hormones which is on TRH, that thyroxine releasing hormone, thyroxine tropical hormone, and then releasing hormone, that's a thyroid releasing hormone. TRH is thyroid releasing hormone. Thyroid releasing hormone. That will stimulate. I'll write it in a different sheets. About that. I'll write it here. TRH. Otherwise, we call it as the thyroid releasing hormone. It will stimulate. It stimulates what? TSH. What is TSH? This is the thyroid stimulating hormone. Thyroid stimulating hormone. Stimulating hormone. And then this thyroid stimulating hormone, which will release what? T3 and T4 release. This one of this mechanism where it is being released. So T3, T4, it is not come just like that. It cannot come, but it needs a stimulation. That's what TSH. And TSH, this stimulating hormone won't come. TRH, that the thyroid releasing hormone. So for one hormone synthesis, you need the another hormone TSH, and for TSH we need another hormone. So finally you get the TRH activate TSH, and then T3 and T4 release. Before TRH, what is that? We also require here the neuroendocrine cell, endocrine cells. That will also, you know, increases the TRH level. So this you need a stimulus package, like, you know, it increases the activity of neuroendocrine, and neuroendocrine increases TRH increases TSH increases T3 and T4, and thereby you get a thyroxine hormones being synthesized. Yes, go ahead. What is that? TRH, which is coming here, releasing hormone, here. This one which is coming here, you go over here. So the TRH, and this is the thyroxine releasing hormone here, the releasing hormone which I mentioned here. So this neuroendocrine cells that will produce on the response of this neuroendocrine stimulus, which is from the hypothalamus, it will put forth on the TRH, because here the neurotransmitters which is coming here. So the TRH which is coming from here, releasing hormone from the nerve endings onto that. This side, this TRH will go, and the pituitary hormone, pituitary cells, the anterior pituitary, and it will bind to the receptors, and thereby it releases TSH. So the second phase is the TSH is released. Okay, and TSH will go, and then it will go where it will go? It will go to the thyroid gland. This will go to the thyroid gland, and the thyroid gland, where already I mentioned before, you get what? TGI peptides, and you know, thyroid glands, the follicles, right? The follicles will produce the TGI, where the iodine is added, and then the TGI is being processed here, and then you have T3 and T4 which is being produced. So this process which is being taken place by the use of what is called the TSH, that's a thyroid stimulating hormone, this thyroid stimulating hormone, it will activate the TG, thyroglobulin synthesis. Once the thyroglobulin is there, and the iodine is added together, and also TSH which help and release the T3 and T4. You follow that? This is a very good sequence of the brain to the anterior pituitary. Anterior pituitary put for TSH, and TSH will go directly onto the thyroid gland where the cells, and it will increase the thyroglobulin to the T3 and T4 and releases. You follow? Yes, okay. So that's the releasing hormones. Another one which we have to remember here is the gonadotrophic releasing hormone, GnRH, which will just call the gonadotrophic releasing hormone. This will stimulate FSH, that is follicle stimulating hormone, and the follicle stimulating hormone will go on the luteinizing hormone LH, and then the LH will now produce on the estrogen, okay, and another way luteinizing hormone activate the testosterone. So we will see that one in a minute. Before that, I would like to, you know, get into the thyroxine again, so that we will complete that study of the mechanism of the release. As I mentioned before, TRH, thyroxine releasing hormone, that will stimulate what? TSH, thyroxine stimulating hormone, okay, and that will end with the release of T3 and T4 as the thyroxine hormones. Okay, where the signal is coming from, I'm recapturing it. The signal is coming from the hypothalamus, or the cold stress, or hypothalamus, or the temperature, you know, those conditions, or any other stress which is coming to stimulate stimulate the TRH release. This is taking place in the hypothalamus. Hypothalamus. Okay, and it releasing hormone. Hypothalamus releases this hormone. So it releases from where? Neuroendocrine cell. Neuroendocrine cell. Okay, and then it stimulate. We call it a stimulation. And then this stimulate. I put a positive for the stimulation. Stimulate. Okay. Now, this thyroxine is stimulating hormone which is secreted in the pituitary origin. This is pituitary. Anterior pituitary, we did study. Okay, pituitary. And these cells will put forth, there are two types of the molecules like a heterodimer. As I mentioned before, the TSH hormone will have alpha subunit and beta subunit. I'm talking about this, how this the thyroid stimulating hormone looks like. It will have two subunit, alpha and beta subunit, and they have a molecular weight of 28 kilodalton. This is the molecular weight of the molecule of TSH. Okay, and they are stored in the vesicles. TSH is stored in vesicles. Stored as vesicles. As we previously did study vesicles. Okay, so they are called. It's not a similar one, so it's a dissimilar. Alpha is different and beta is different. Both together, if you form 28 kilodalton, so it is called a heterodimer. Okay, this is the type of protein. We call it as a heterodimer. If both the chains are alpha and alpha, so two alpha, maybe that's called the homodimer, but you get the heterodimer because alpha peptide and beta peptide is different, so we call it as heterodimer. So we are talking about here the thyroxine stimulating hormone. It contains alpha and beta subunit, and the molecular weight of TSH is 28 kilodalton, and TSH predominantly secreted in pituitary gland, and this TSH is stored as vesicles. So whenever there is a command to release, yes, by the TRH, then the stored vesicles now it's being released into the next level, into the blood. Okay, this will TSH now enrich more amount of TSH now. This will stimulate more of TSH to T3 and T4, and this one will go to the body. Now it is secreted in the body, and where it will go in the body? Into the cells. Inside the cells, where it will go, it will act in the DNA regulation, DNA gene expression. Okay, go to the DNA gene expression. Now, you cannot have always increasing T3, T4. You want to shut off this mechanism. So what the mechanism here to turn off this, this T3, T4 is a feedback signal. It will stop the TSH. There is a level which our body can require the need for the gene expression. Once the T3, T4 does its function, after that there is no need. So excess of T3, T4 will go and stop TSH production here, gene expression of this gene in pituitary gland. Because this is in the body, right, in the blood. And then once the level is gone up, then it will automatically go to the pituitary gland and stop the TSH production. The T3, T4 will negative regulation. At the same thing, it will also do a negative to thyrotropic hormone in the hypothalamus region, releasing hormone. So it adjusts itself like a feedback mechanism. Feedback. What type of feedback is it? It's a negative feedback because it inhibits, so we call it as a negative feedback mechanism, this one, and thereby it stops. So it stops where? TSG gene. TSG gene is being stopped. It will go and bind into the DNA of the TSH, and then it stops. Do you follow now how the T3 and T4 is secreted? And once it is secreted, how it automatically switch off its own synthesis, like a feedback mechanism. So the TRH and TSH and T3 and this, as you mentioned before, it will bind, and how it will bind into the cells, TRH to TSH, which we already studied about GPCR. Have you studied the G protein coupled receptors in the last class, and also IP3 and calcium mechanism? So during the GPCR activity, this TRH hormone bind with the coupled G protein coupled receptors, because as I mentioned before, and this hormone which is having its membrane receptors in the membranes, which we studied earlier for the steroid hormones like GPCR mechanism. The same mechanism holds good for the thyrotropin releasing hormone, and here the TSH to T3, how this been activated, the same mechanism, GPCR mechanism. Also it will also induce cyclic AMP, also IP3 calcium. So these are the mechanism of binding, of how the TRH is binding to the membrane, and then it will activate TSH release. Now TSH release, how it will activate T3 via GPCR binding mechanism, and thereby it turn on the T3 T4 release. So once the excess of T3 T4, we don't want that excess, and then it will go and shut off that. Because what is the mechanism which I mentioned before? We have a cold stress. If you are in a cold, or if you are having a starving always, I mean, you don't eat anything. And that stress in turn give a signal in your brain, I am shivering, I am cold. So that kind of stimulates the TRH release. Then TRH will stimulate TSH, TSH T3, T4. Then after that, T3 and T4 will go to the body and activate into the mitochondria, and thereby it uncouples the oxidative phosphorylation, where the energy is being converted into the heat energy, so your body become warm. Okay, so your body is having your own automatic like a thermostat. So the thermostat regulation is buying T3 T4. It regulates. Once the body temperature rises up, after that there is no need. We don't want to go into the like a fever. No, you want to have optimum temperature. So at that time, the T3 T4 excess will go and turn off this gene, and thereby we are maintaining the T3 T4. If you give excess of T4, T3 T4 or thyroxine to the stress, then you are giving unnecessary burden to the body that induces certain type of pathological abnormalities. So one of the experiments we did in the early days, we inject a thyroxine to a rat. Not normal amount, excess of. Normally that will grow. If you go excess, what will happen? You get extra work. As you said, the body will do, the mechanism of thyroxine will increase the metabolism. If you increase the metabolism, you need oxygen. If you need to increase the oxygen, you increase the blood output. So if you increase the blood out, your heart rate will go up. So when you do, the heart rate will go up, you are unnecessarily stimulating the heart muscles, so your heart will become hypertrophy. So hypertension and hypertrophy will go hand in hand. So thyroxine at a high level, unnecessarily, that will induce the cardiac hypertrophy. So that's what we did in the heart. Then we did in the fish. What we do? Inject thyroxine to the fish constantly, every alternate day. What will happen? The fish will uncouple in a gill's mitochondria, and that stores excess of the calcium, and thereby it uncouples. So we are inducing the fever in the fish. Okay, and then if you take that fish after thyroxine treatment, and then we see that how the mitochondria is being activated. It's highly activated. Those are our own research in the early days. So we did with these thyroxine many mechanisms in our body. We increase the metabolic rate. So in the clinical conditions, which one of the things we call it as the hypothyroidism. Have you ever heard of that? Hypothyroidism, where the swelling of thyroid gland. Okay, if this is a thyroid gland which is coming up. So normally the thyroid gland, this is the tracheal, and these are thyroid gland, and this is going swollen like this. Thyroid gland which is swollen because you are not eating enough of iodine. So if you eat iodized salt, that's good. In a mountain range, the people they suffered here because they need the less amount of oxygen when you go on the higher up. Also, they don't eat much of the iodine or whatever the iodine which you eat. They have to cope up with the cold stress. They have excess need of thyroxine, but you are not supplying enough amount of oxygen and enough amount of iodine. That induces unnecessary stress. So you have a swelling in the thyroid gland, and that leads to hypothyroidism, where the less amount of thyroxine in their body, and that will give into the low metabolic rate, low metabolism, and those people are very lazy and they cannot do any other work and keep quiet, and not much on those. Okay, that's, I don't want to go in detail because that will lead to a pathophysiology class, not the comparative physiology class. Okay, so in the thermoregulation, which we just now we discussed, thermoregulation of these brain signals, okay, which will give rise to what? T3 and T4, okay. And as I mentioned before, T3, T4 will go into TSH with negative feedback, right? If you excess thyroid stimulating hormone. In a positive feedback, in a positive way, whenever the cold stress, it will give a positive, that through the TRH. TRH will activate the TSH, and then TSH which will go into the T3 and T4. And it will go where the targets for the T3, T4? Body, right? What body? The liver, kidney, heart, and nervous system, and skeletal growth, skeletal membrane to increase more of the calcium. So if you increase these T3, T4 or the thyroxine hormone, okay, in a genetic level, and it increases the person's height. So when I see this particular picture, could I see this guy? He's a tall guy. So what do you think this guy's T3, T4, and compared to this girl? Tallest person, Robert Pershing Wadlow from Alton, Illinois, reached a staggering of 2.72 meters by the time he died in 1940, age 22. Okay, he also got the world record on this guy. So that's been increasing this level. And then opposite here, you can see this guy. What is this and this? Excess of thyroxine level, T3, T4. And this guy is the shortest person. In 1990 in India, Gul Muhammad was crowned the shortest mature man in medical history, this guy. He was a mere 0.57 meter tall. And this guy, 2.72 meters tall. Imagine, that's everything on to do with thyroxine. And another one is the neck, which is the region where you will find the thyroid gland is there. And this is the one of the longest neck, but this is nothing to do with thyroxine. But just I want to show you for your fun. By wearing a succession of copper coils, women in the Padang tribe of Burma have extended their natural neck. If you have a short neck, if you want to increase your neck height, you can use these copper rings onto the neck, and that will increase. That's what they're feeling, and now they got it. Okay, that's one of the things with the new scientist. Okay, the next part is what we need to study, the iodinated tyrosine. Iodinated tyrosine, otherwise we call it thyroxine. Okay, iodinated tyrosine. Tyrosine is an amino acid. Iodinated tyrosine is thyroxine. It exists in a two form, T3 and T4 forms of thyroxine. Okay, they have not only in the human, you also it regulate in the developmental growth of jellyfish. It regulates, as well as in sponges. It regulates the growth of this organism. Okay. Now let's go on to the the production of testosterone, another hormone. Okay, you will study about testosterone. So we call it the male hormone. We call it like that, testosterone, and it is mainly synthesized from like a LH, luteinizing hormone, and follicle stimulating hormone, just like a TSH in previous case, which we studied as a thyroxine stimulating hormone. See, testosterone is stimulated by luteinizing hormone, and FSH is a follicle stimulating hormone. The site of production, where is the site? Site of production of testosterone, where? Site of production is testis. In the testis, it is in the seminiferous tubule. Seminiferous tubules. So seminiferous tubules are the site of production of what? Testosterone. Now we will see the regulation of this one, of this the exact site and everything. Okay. So in the testis, okay. So you get the follicles. You call it like this. And it's just several tubules. There are several tubules are there. Seminiferous tubules. I'm just drawing there. These are the seminiferous tubules which is present in the testis. Okay. So they are called as semini, seminiferous, F-E-R-O-U-S, seminiferous tubules. T-U-B-U-L, tubule, L-E-S, tubules. So it has its own cells in the tubules, okay? It's kind of concentric rings like that. It comes out like, you know, another cells will come like this. See, like this. These are the another cell, and then another cell, another cell. So it looks like this. It comes out. So the mature cells over there, and then the they are called, these cells, we call it spermatocytes. These are called spermatocytes, C-Y-T-S. Spermatocytes are the cells which is present in seminiferous tubules. Okay. Now, these cells are matured. When they mature, these spermatocytes will produce sperm cells. Sperm cells. How it mature? By meiotic or meiosis. Spermatocytes that undergo the meiosis, and that will produce sperm cells. So when they mature, it will travel inward. So you get these sperm will be produced over here. These cells will produce more of mature cells will come at the middle, and you get the sperms will be running around at the middle of the seminiferous tubules. So when these cells are there, I'm writing the same seminiferous tubules. The another type of cells, see, as I mentioned here, these are the cells, okay, the same thing, spermatocytes. There's some cells over there here, another type of cells, okay, along with that, like that. So it also here is going to be here. And these tubules, or these cells, we call it as Sertoli cell, S-E-R-T-O-L-I, Sertoli cells. And this Sertoli cell, what these cells will do, it will nurture the sperm cell. Nurture will provide food, right? Nurture sperm cells. Why? Because they are meiotic division. This is not a whole cell, not one cell, but it's a half of the chromosomes. So it cannot perform its own function. That's very important. Sperm cells, they are half of one particular cells of the sets of chromosome. It cannot live on its own. It requires more additional. So the Sertoli cells will provide some of the help. So they nurture the sperm cell to be mature. And these cells are present in between. Look at this one. In between the spermatocytes. Okay, in between, and also in between the tubules over there. Okay. Along with that, okay, you get the Leydig cells, some of the other cells outside the seminiferous fluid. These cells, which is present outside of the seminiferous tubules. And these cells call as, they call it as a Leydig cell, Leydig, Leydig cells. Okay. They are present where? In between the seminiferous tubules, right? These cells are present, this is one tubule, this is another tubule. In between that you get these Leydig cells. And these Leydig cells will produce testosterone. Okay, Leydig cell produce testosterone. So you should remember one. In the testis, you have seminiferous tubules, these tubules, and they have spermatocytes inside. And the spermatocytes, when it matures, it will travel at the middle and it will produce more of, by meiosis, to produce more of sperm cells. And the sperm cells are the middle, but it is a half of this chromosome, so it is not, it's a haploid, and it is not like 23 pairs, but you have 21 chromosomes, not a pair. So it requires certain other things to survive. That is being produced by the serotoli cells, these cells, which will nurture the sperm cell, so the sperm cell will be alive. So sperm cells is being produced. Now, in between the seminiferous tubules, another type of cells, we call it as a Leydig cells. These are the Leydig cells, or Leydig cells, and these cells now produce testosterone. So you have the male hormone, or the testosterone is how that's being produced. Now, how this testosterone is being stimulated by LH, or luteinizing hormone. Okay, do you follow now? How that is testosterone is going to stimulate, and I'm going to get the next mechanism how that is going to happen now. Okay. In the gonadotrophic releasing hormone, GnRH releasing hormone, as I mentioned before, whenever I say the releasing hormone, it should come from where? Hypothalamus, in the brain. Hypothalamus in the brain. Okay, so hypothalamus, from that the neurons and everything that releases the GnRH. Okay, and this hormone, it is a peptide hormone. How many amino acids? 10 amino acids. Okay, gonadotrophin releasing hormone. Okay, it is a peptide hormone. Okay, this hormone now will stimulate, as I mentioned before, two things. One for follicle stimulating hormone, FSH, and another one, it stimulates luteinizing hormone, LH. It stimulate, positive stimulate. Luteinizing hormone will go to Leydig cells, Leydig cells, Leydig cells. These Leydig cells now, as I mentioned before, last slide, it produces testosterone. Okay, and testosterone what? It is a steroid hormone. Now, the testosterone now, which is again as a negative feedback. Okay, you don't want to have testosterone always being produced. We need to shut off that one. Okay, so this testosterone now, the excess, which will inhibit one. In a case directly related to the follicle stimulating hormone, it negative feedback. Another one, it can also negatively regulate, or negative feedback, it stops the production of the gonadotrophic releasing hormone, negative. Okay, so positive, it gives a luteinizing hormone, and then it Leydig cells is a positive, means it stimulate, and then it will output is testosterone. That's a steroid hormone. If excess of steroid hormone is not necessary in our body, so whenever the testosterone is necessary, it will produce, otherwise it keep quiet. So we have to have a feedback mechanism. It's a high level. It will stop the follicle stimulating hormone 1 and another only gonadotropic hormone. It stops over there. The follicle stimulating hormone, just like the Leydig cell, it will also output to, it will stimulate what? It will Sertoli cell. Which I mentioned before? Sertoli cells? Yeah. Sertoli cells. In the Sertoli cells, it will produce inhibin. And this inhibin, now inhibin, INHIBN, inhibin is a hormone. And this hormone, which will, just like Leydig cells, the testosterone, but inhibin will produce, and it will inhibit this hormone, follicle stimulating hormone. It inhibit its own. So if it is an excess about, yes, follicle stimulating hormone, it will go on Sertoli cells and then inhibit. So in thereby, this is being controlled. What the Sertoli cells will do, it will nurture the sperm cells. So we don't want to get the sperm always being done, so it has to stop. So the Sertoli cells is being inhibited by the inhibin, okay? And that is from the testosterone in one side, the follicle stimulating hormone. The other way, it can also stimulate the gonadotropic, the FSH level. Will determine, okay? So, sorry? Negative feedback. Sorry? Sertoli cells will put forth on the inhibin. In a normal way, it will produce an inhibin level, okay? So when the inhibin is there, then it will inhibit the FSH, and thereby it will control the Sertoli cells output for nurture the sperm cells. Right? See, whenever the testosterone, you stop it, but you are not stopping on the sperm production as well as the sperm output or sperm survival. So that is being again negatively feedback controlled by FSH, or follicle stimulating hormone. So here you have to remember the releasing peptide, or the gonadotropic releasing hormone, is a peptide hormone or protein hormone which consists of 10 amino acids, but that releases a steroid hormone. So one hormone is releasing another hormone. Okay, follow? Now, we'll go on to the targets of all these hormones in a way that we have to study about. The next one, we have to study about estrogen and progesterone. Otherwise, we got the LH and FSH. So luteinizing hormone as well as the follicle stimulating hormone will also responsible for producing the estrogen in female. Okay? So otherwise we call it like that, the symbol. Okay. So in the ovary, where exactly estrogen and progesterone in the female is the ovary. Okay. So what happens in the ovary? There are two phases in the ovary, and even when it been activations, and there are the stages. We call it as the pre-ovulation. Ovary. What the ovary will do? Ovary will produce more of ova, right? The eggs. So pre-ovulation. Otherwise, we call it as a follicular phase. Follicular phase. FOLLICULARPHASE. Okay, follicular phase. Pre-ovulation, this is number one, or the follicular phase. Another one, which is you need to remember, that is on luteal phase. Lutea, luteal phase. Second phase. This is called a post-ovulation period. post-ovulation phase. Hello? Anyone? Any question? Okay. Now, let us see how this estrogen is being produced. Okay. The ovary, I just draw structure of an ovary like that. Okay. In this, as I mentioned before, the first one is the follicular phase. Okay. Where you get the more of the cells, is immature cells in the ovary. They migrate surrounded by oocytes. We call it the oocytes. oocyte, oocytes. Okay. So this is the cell. Okay. The cells, that's a thick one, and the oocytes surrounded by oocyte. The oocytes are here, like this. So they are oocytes. Okay, the cells, and these are the follicles. Is coming up, and then it grow in the follicular size. It increases the size now. Increase more of the oocytes. So I write like that. Increase in size as it mature. More of oocytes over there. Okay. Then after that, what happens is matured oocytes. It's a big one here. It is being larger size, and some of these oocytes is over there, and that is going. This large one is being released. This is the releasing part outside the ovary. Okay, and the rest of the things which is not coming out here, it will form like a corpus luteum. This one is a regular corpus, corpus, corpus luteum, luteum, corpus luteum. Otherwise we call it a CL, corpus luteum. This is what we are talking about in ovary, what happens. Okay. So the first phase, with the follicular phase, the migration which is occur whenever, and selection and growth and migration here at this phase. Okay, what happened? Selection of good cells, selection, growth which is occurring of the cells, and migration. So migration of the cell. So the oocytes, O, capital O, small o, oocytes, okay, surrounded by follicular cells, follicular cells. So these follicular cells surrounded by oocyte, many oocytes which is going to be in there. And then the follicle cells that produces, these cells will produce estrogens, estrogen and progesterone. Okay. And the follicular cells, what these follicle cells are, write it in a separate paper. The follicle cells, they are the endocrine cells. Endocrine cells. And that produces what? Estrogen and progesterone. Okay. Also, the follicle cell also will produce, these endocrine cells also produce luteinizing hormone and FSH, follicle stimulating hormone. They will produce. You follow now the process how this from ovary there are different cells and oocytes, and the follicles is responsible for the synthesis of the FH and FSH. Now, how it will act, the mechanism we will do. Okay, the release. As I mentioned before, gonadotrophic releasing hormone, GnRH, okay, the hypothalamus, that will increase a luteinizing hormone through the follicle stimulating hormone, right, LH, and also it will produce FSH, follicle stimulating hormone. And this hormone again in turn positively feedback, positive. It can act on follicle cells, and it will produce more of estrogen and progesterone, PG, I put it in a short form, and then it will also regulate the progesterone, will also negative as well as the positive, that depending upon the level, it will activate the gonadotrophic hormone, estrogen. Okay, the early phase, we have a different phase, we call it as a follicular phase as well as the luteal phase. Now let us see what happened in the follicular phase, okay? Follicle phase, follicular phase, okay? There are three types. One is called the early, another one is the mid phase, another one is the late phase. Okay, there are three subdivision here. And the early phase, what happened here, the low level of FSH, follicle stimulating hormone, okay? If it is a low level, what will happen, it will the follicle stimulating hormone, you get the low of estrogen. Is that right? If it is the low level of this estrogen, it also act on pituitary cell, okay, and it will produce the low level of FSH in the pituitary, low estrogen in pituitary. So it is a negative feedback again, negative feedback. I'm talking about the early phase. The early phase, there is a low level of follicle stimulating hormone, so low level of estrogen, and then that in turn will also negatively feedback. So this low level will be maintained by this negative feedback. Okay. So it act on the follicle receptors, follicle stimulating hormone receptors. So that's the level it will activate. The next phase is the mid phase, when there is an increased mid phase, increased LH, luteinizing hormone, and FSH. Okay.
Dr. Solomon?
Yes, I will write it in a different paper. Right.
I have a question.
Yes.
The early part of follicular phase, you said
Yes.
Low levels of estrogen will in turn produce low levels of FSH?
Yes, that's right. There's a negative feedback because the low level of estrogen will activate in the pituitary gland, and then that will produce in the low level of FSH again. So it will maintain the low level at the early phase, it will be maintained. Now if you go on to the follicular phase, mid phase, what will happen? Let us see. Okay. The mid phase, you have an increased LH and follicle stimulating hormone. This will activate, okay, the FSH, follicle stimulating hormone receptor, FSHR, and LHR. It will activate, this hormone will activate the receptors, and this will give rise to increased estrogen. The increased estrogen and progesterone, and thereby you get the increased LH and FSH again. It will have a feedback mechanism again. It will have a positive feedback. So if you have the action is the increased LH in mid phase, we got mid phase, or the follicular phase mid phase, LH and FSH activate the FSHR and LHR, the receptor is being activated. When these receptors are activated, that will increase the estrogen content, and if you increase the estrogen, that will also increase. So it is an increased phase. Now the next stage is the late phase, the follicular phase. The late phase, what will happen? This will give, a positive feedback. This is the only positive feedback. If you have more of LH and FSH in the mid phase, what will happen? Increased amount of estrogen, increased again LH, and then increase. So it's always you get an increased level. And this is, we call it as a hormone surge, S-U-R-G, hormone surge. That means this hormone, excess hormone, is more hormone, So this will lead to ovulation. See, ovulation is not taking place on the early phase, But due to the mid phase, you get there is hormone surge because of a positive feedback inside these receptors estrogen, and then increase, and then suppose if you have a 5 molecules initially, it activate another 5 molecule. But this go and activate another 5 molecules. So this 5 will get 10, and then here the 10, another 10. So if another round of 10, you get a 20, another 20 will come out of 40. So you get this more amount of these hormones being produced. So that is, we call it as hormone surge. If you have excess hormone, that will lead to ovulation, and that will give you, during the ovulation, what will happen? More of the estrogen and progesterone is being produced. Is that right? That is just writing on a separate paper. So during ovulation. During ovulation, what happened? You have a more or excess of estrogen and progesterone. This hormone. Excess. Okay. And once it is excess, then we don't want that excess once the ovulation is occurring. Okay. So what will happen if it goes on the luteal cells? If it goes on the luteal cells, it will produce in a corpus luteum. Okay. And that is they are present in the endometrium of the uterus. Endometrium of uterus. Okay. So this LH, now we are going to talk about the after the ovulation is occurring, we are talking about the luteal phase, the next phase, not follicular phase, luteal phase. Luteal phase. So that's what it happens. Okay. So once this ovulation occur after the follicular phase, then the next phase will start, that the luteal phase, where the LH hormone, luteinizing hormone, that give a negative feedback to the estrogen and progesterone. Okay. And then these estrogen and progesterone, okay, that will also produce onto the negative feedback and the excess if you go for luteinizing hormone. And this will go onto the negative. So negative feedback, once it is being done, we need to stop. So through the luteinizing hormone, this is being regulated. Estrogen and progesterone is being regulated. And once this is going down, so you get the endometrium, you get corpus luteum, that is going to reflect, and then that is coming out the CL, or the corpus luteum, that is being removed. So we don't want excess of those, the ovulation part. So it stops, and then if you have produce of one of these ovas being produced, and that is released into endometrium of the uterus. So that's the one part, how the ova is being released into the uterus and ready for the fertilization now. So we will see the mechanism of the release. I'm summarizing up now. Follicle stimulating hormone, that will produce more of the progesterone. Okay, that is a positive feedback. And also FSH will also produce, when it if it drops, you get what? If FSH drops, luteinizing hormone, and the luteinizing hormone, the corpus luteum, and that leads to degeneration. So that is a cyclic period of this corpus luteum and the decrease, and that's the part of. And then the progesterone is then endometrium. If it is decreased, progesterone decrease. Progesterone, if it is decreased, okay, and then you get the endometrium is degenerate. Endometrium degenerate. All right. So if the follicle stimulating hormones, if it is a low level of LH, you have a corpus luteum is degenerating. If a progesterone, and if that level is going down, you have the endometrium is becoming degeneration, which is occurring. Okay, now you remember this topic now. I'm going to get into why fertilization is occurring or no fertilization. If there is no fertilization is occurring, what will happen? No fertilization. no fertilization. So you get no fertilization and no progesterone in endometrium. Okay, endometrium. So if no progesterone in endometrium, what will happen? That will degenerate. Right? That's what I explained here. Progesterone level decreased, endometrium decreases. Okay? So no fertilization, you get no progesterone in the endometrium, and that will lead to degeneration of what? Of the ova. That's gone up. Now, if there is a fertilization occur, if the sperm is there and the ova is there, and fertilization is occurring now, fertilization. If there is a fertilization, what happens in the uterus? In the uterus and the uterus endometrium is not degenerating, but it will produce a hormone like a chorionic gonadotropin hormone. It will lead to CHO, chorionic, CHORIONIC, chorionic gonadotropin hormone. It will produce a chorionic gonadotropin hormone. Okay, and this will produce of corpus luteum being activated, and that will again progesterone positive. It will increases progesterone release, and it will help the progesterone help the uterus endometrium help to develop. Now the fertilized egg will get enough nutrients and everything, and the healthy endometrium, and thereby that particular embryo will be developed after the fertilization. That helps by progesterone. So because of this uterus endometrium, that produces more of chorionic gonadotropin hormone, and this hormone will activate the CL, that is the corpus luteum, and then they keep more of progesterone. More progesterone will help uterus endometrium, and this process is going on. So the corpus luteum, I mean, that's also take input from the brain. It will also take the input from the brain as well. Hormone is a positive one. So if you have progesterone, okay, progesterone analog, what will happen if you have progesterone analogs of excess of progesterone? That's if you take progesterone, which will help what will it do? The progesterone, if you excess of progesterone, that will help in the endometrium, and that can also prepare the uterus to receive the embryo, right? And also it will act on the hypothalamus pituitary, as I said, the brain, and it will also activate, to production of GnRH, gonadotropin hormone. It's a feedback inhibition, okay, and a follicle stimulating hormone. Okay, so without LH, so this progesterone analog, if you have excess of progesterone, you are supplying as a birth control pill. Have you ever heard of that mechanism? Anyone? How the birth control pill will work on that? Yes, Salman, you know that? No? Okay. I mean, you put the progesterone analog, analogs, and they are as a birth control pill, contain the progesterone analog, progesterone, and the body thinks, okay, if you add more, body thinks, what? Body thinks? Yes, you are. It is pregnant. So if it is pregnant, what it will do?
Stop ovulation.
Correct, stop ovulation. That's what I want to know. See, when it fertilized egg, uterus, endometrium, chronic CL and progesterone is more, and that will help develop an embryo, right? But what will happen when you take of the artificially made or progesterone pill or the birth control pill? If you take excess, there is no fertilization, no egg, okay? So what happens if you take excess of this? So this will think, the body think that it is it's pregnant. So and then what will happen? It will stop ovulating, and thereby there is no embryo will be developed or no fertilization will occur. So this will inhibits the development of oocyte, because it stops by inhibiting FSH and LH. Do you follow? Birth control pill. Excess of progesterone that will mimic as if the body thinks that he has pregnant, and then stop ovulations, and thereby it inhibit the development of oocytes, and thereby the low level of FSH and LH is producing stop. So you are manipulating the hormonal surge and the hormones in our body. So that's the part. Sorry? Yes. It will. It thinks what will happen if it is a pregnant. Rest of the body normally the further development of FSH and LH is being stopped, and more of oocyte, the which I mentioned before, the early phase and then the late phase, and those things will not be taken place here. So it stops the follicle cells, right? See, I'll give you here. See, this is the one, right, where the ovulation which is taking place. So when this oocyte is forming, right? So for this process, is the normal is fine, where there is not much of progesterone, okay? But estrogen is there. So here, if you see, this oocytes is being developed and the ova is being produced, and that is being released, and it will take the days of time, right? Is a period of time. So when it is not being fertilized, it is going to be waste. It is going to be endometrium is degraded, and that is going. That's a normal cycle of that one. Suppose if there is a sperm there and it fertilize and it produces more of the embryo, it needs to be feed and health. So at that time, it turn off the ovulation process. At that time, you have an excess of progesterone in our body. In the body life. So if you think, you make the body to think that it is pregnant, you are giving excess of progesterone, give more progesterone to the body in a normal, healthy individual. But at the same time, you also, to stop the ovulation, that in feedback mechanism by stop the ovulation process, because the body think it is a pregnant, and not to, synthesize more of the oocytes and more of follicle and more of ova, and thereby. So it is a initial stage, you need the progesterone, estrogen to release the oocytes, okay. But after that, once the pregnant is there, at that time you have excess of progesterone level is being there. So the people, they think, okay, I will supplement with the progesterone analogs as a birth control pill, and thereby stop ovulation process. So that's the mechanism. You follow that? Still you don't? Any question? Okay. So what I'll do, I'll give a break now, and then we will do a development of the insects with the different hormones, and then we will review for the exam for the next week. Okay? Review. Okay, now we have the development of insects, because it's a comparative physiology class. I have to compare with the other organisms as well. Okay, so the insects of the development, you know about the metamorphosis, right? Metamorphosis. Metamorphosis is, what is metamorphosis? From the egg laying, and then you have a larva, and then larva to pupa, and pupa you have an adult insect which is coming out. So in the humans and other organisms, the metamorphosis is a bit different. Like if you see the egg and fertilized, you have a development and then you are coming out as an adult. But in the insect, there are different stages in the larva 1, larva 2, larva 3, and then pupa, and then going to adult. So the hormones, they play a major role in the development of insects. Okay. Also, they call it as molting, which is occurring in the insect. And the insect also, they have a exoskeleton. Insect, they have exoskeleton. And they call it as this exoskeleton, we call it as a molting process. Molting. Okay. And this molting is by sexual maturation and by hormones. Okay. And in terms of this, in hormones, we will study the what hormones there. Okay, hormones that in the larval stage. So larval stage, the hormones are there. When the adult, the hormone is not there. So the development that depends upon the hormone. What type of hormone? Let's see. In the insects, hormones responsible for shed to grow of exoskeleton. It induces a shed to grow exoskeleton. That meaning this process we call it as a molting, which is coming up. It produces a complete metamorphosis. Okay, it occurs in beetles, okay, flies, bees, wasps, okay. So all. So what happened? The 90% of all insects of the planet, 90% of the insects on all planet, we have got 50% of species in the whole species on the earth. 50%, mostly 50% of species are from the insects. Okay, so the embryonic stage, the caterpillar or larval stage. How many of you have grown tomato plants in your yard? Have you seen caterpillars coming up? When the caterpillar comes out, what happens? It eats all the food, all the leaves, all the green leaves. Caterpillar comes out. So this stage is the feeding stage phase. That meaning it's the embryonic development. Still, it is not an adult, so we call it as an embryo. So in embryonic development, it consumes a lot of food. And then it will lead to a pupa stage where it won't eat at all, right? It became a cocoon-like, pupa stage. And then it is being released in a different form as an adult. Okay, so that's why I call it as a metamorphosis stage. Okay, in the development there are two types of hormones. Call it. Two hormones responsible for this. Two hormones in the development of insect. Call it ecdysone. 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 Another hormone, they call it as a juvenile hormone. Or we call it JH. So it looks like a steroid hormone. Looks like this. The structure of this hormone, ecdysone. Steroid neuroendocrine cell. Okay, steroid hormone that produces from the neuroendocrine cell. Neuroendocrine cells. Okay, and it will release a brain peptide hormone. It releases neuroendocrine cells. Okay, this is a steroid hormone, the ecdysone here, And the mechanism of release is the neuroendocrine cells in the brain that output a brain peptide that is called PTTH. The peptide name called PTTH. That is called, this name called protothoracic tropic hormone. PTTH. Okay, PTTH. And this hormone now onto the gland. I'll write on another sheet. PTTH. Okay, this is a peptide hormone that will act on glands, and that's the endocrine gland. That the gland now secretes what? Ecdysone. Okay, and the another one is a juvenile hormone. Okay, juvenile hormone, and that is again is released from endocrine cells. Endocrine cells. Juvenile hormone is also synthesized, and both of them, these ectodyne and the juvenile hormone, okay, and they have in blood, the in the I think in the hemocoel, it is not in the insect, it is not have a red color blood, but they have got a liquid, right, colorless liquid, and they also have a membrane soluble, this one, and they also have cytosolic receptors, cytosolic receptors, membrane, the hormone receptors. This behaves the same thing like a steroid hormone, and they also have a blood carrying protein, blood carriers for this hormone. Okay, so the mechanism is the same, but it will act in a different way. This is the mechanism of release of the PTTH on the gland, and gland is ecdysone, and juvenile hormone the endocrine cell will produce, and both of them on the blood, and they follow the same track path of the steroid hormones. Which one? The last one here? This one? Okay. JH and ectodyne in the blood or in the in the liquid. We call it, in the insect they won't have a blood, but they have a hemocoel, like a liquid, white color liquid. When you drive in a in a car and you get all flies is coming out into your windshield, and windshield become full of insect blood, right? We call it. It's not a blood, but it is a blood, but we call it as a blood. It won't hemocoel, the white color liquid. But wherever it feeds on all the pollens and then other whatever sometimes if they feed on some other leaves, some other insects and everything, that insects fed on some fertilizers and herbicides, you get all the herbicide smells over there, all these insects or insecticides perhaps. So that comes up. Okay, now the stage is an interesting part. When in the larval stage, okay, day 1, 2, 3, 4, 5, like that it goes on. So it increases the hormone, the ectodysine, and then it decreases whenever it goes up. So it's a bit of a complex one, and then it's a day second. So whenever it's a surge of excess of hormone, what you get really, that insect is developing at the different days. Okay, 5 days, and then it goes on, and then in the pupa, this is the larval stage. Larval stage, every day you get this is an increase. I'm drawing the level of the hormone ecdysone. So it increases and decreases, and then a particular time of day increases. So in all the larval stage it increases, but when it reaches the pupa stage, it goes up and then it goes slowly, and then it increases that one, and then it increases, and after 18 days it goes up and decreases. In the adult phase is nothing, it is not expressed in the adult. Okay, and if you take care of the JH level, that's on the juvenile hormone, which is going to be steady. It is not increasing at all, it's just steady, and then it stops, and then for up to certain days or 14 days or so, and then it slightly it increases, okay, and then in the pupa is nothing there, in the 18 is nothing's happening, and then in the adult phase, this is an adult one, it increases, okay. So juvenile hormone and the ecdysone hormone act alternatively in the developmental stage, and then you will find in the adult no ecdysone is being out, but juvenile hormone it increases in the adult phase. So it goes like this. Here you can have the no JH, but here the 18 days what you have more of the ecdysone during the pupa stage. It is there, here it is there, but there is nothing is there, okay. So it is the JH plus this is the ecdysone in the larva stage development, and that leads to larval stage expression, and then no JH, no juvenile hormone, okay, no, nothing there, but only the ECD, that in the larval stage, and that will get into the pupa. Pupa you have the ECD, and then the molting is regulated by the molting, which I mentioned before. The molting is regulated by ecdysone. So that's why the pupal stage, the ecdysone, and here the molting which is coming up, you get, but the juvenile hormone is nothing to do with that molting. So in total, the summary of this class, what we have covered in the hormone lectures, right? We did study multiple hormones. We did study the glucose and glucagon and insulin and peptide hormones and then catecholamine and thyroxine and follicle stimulating hormone, testosterone, and then the insect development, ecdysone and juvenile hormone, all of them. And we have studied of the different biochemical pathways. We did studies, different biochemical pathways. And then we also studied, the hormone sensitivity, how the hormone is released, hormone sensitivity. Also, we did study about the hormone regulation and the target of cells, okay? How that is being low concentration and the higher concentration, how they stimulate and then it inhibits the activities and everything. And the target cells, what they will do, their cell decision, whether the cells will synthesize a particular function or not, that is being decided by the cell decision due to the hormone presence, okay. So this will give a comprehensive approach of all the hormones, whatever we studied in the class. Any questions so far? Any question? Yes, go ahead. No?
No.
No question? Okay. Here? Any question? Okay. Now, I would like to go for the exam, midterm exam or exam 1 and 2, okay. What I'll do, this exam is going to be next week, or next time means after the spring break, when you come back, you have a written exam, and you have a multiple choice question, MCQ, and that is from the lectures, part so far we covered here, the class lectures, okay. And then the second part is the short answer questions. Okay, I'll give you now. And then third part is the table. In the textbook, the tables are there. I will give you certain tables. You go through and study that table. So you will get the questions from that as a matching. So you have a short answer and multiple choice as questions and quizzes like type, and then tables. So you have a matching or quizzes from that too. These three parts. Okay, and this is going to take place at 10 to 12 p.m. 2 hours of your exam. Okay, so you have to bring a scantron. Bring scantron to the class. Okay, I will give you a sheet of question paper, and you have to answer in that question paper, and your answer should be there in the answer question. and it is a closed book exam too. Okay, it's a closed book exam. Follow? Okay.